Thank you very much indeed, and thank you very much to biocrates for inviting me to speak here. I'm going to talk about cohort studies in the digital era, and I want to sort of zoom out now to think about how we might change the way we do epidemiology in the digital era using time series tracking, experimentation methods within cohorts. And I'm going to talk about some of these within TwinsUK and also beyond. So this is just a rough kind of like 4 points of my talk today. I want to sort of give you a background about 30 years of longitudinal multi-omics that we've done within TwinsUK. Many of you have collaborated with us. We've produced a huge number of papers, about sort of 700 papers on many different topics, trying to keep ahead of the field at all times. And then sort of a few years ago, we started introducing experimentation into cohorts. And then, of course, the pandemic happened. And the pandemic opened doors for us in a number of ways, which I'm going to tell you about. And this leads me on to sort of think about how we might change the way we do epidemiology. Now we've got a number of new tools up our sleeves. So here's TwinsUK. We started back in 1992, so it's now 30 years on. We've just done our sort of 30-year celebration with the participants, who are incredible individuals who've really stayed with us for the long haul, going from looking at the genetic architecture of lots of different traits, introducing concepts like epigenetics, metabolomics coming in sort of around the middle of the 2000s, moving on to looking at things like expression changes in relation to aging, phenotypes, and then microbiome. Then in the last sort of 3 or 4 years, introducing dynamic phenotyping. And this whole process, as many of you I'm sure will experience within cohorts, it's really a collaboration between the study participants, incredible operations team being willing to sort of get involved and engage them, and a massive worldwide network of researchers who use our data. So it's been fantastic. Now, this is sort of like some of the range of some of the samples that we have developed over the last 30 years, and we're using them with the scientific community and sharing the data from them to really keep ahead of multi-omics across multiple organ tissues to try and study aging. And so here's the biobank facility that we have, which is within King's College London, but also we have sort of samples stored in other biofacilities elsewhere, and it's getting bigger and bigger all the time. And we have a dedicated clinical facility where we've always seen people and visited. They've come to visits every 4 years or so, where we collect a range of data. We've just got funding for a really fantastic imaging to do whole body MRI imaging in the TwinsUK cohort, so to provide another really crucial endophenotype to relate to our multi-omics. And so here are some of the sort of markers that we've done, like many of you in cohort studies, trying to understand aging and aging traits and how they change. And about 5 years ago, we instituted some interesting dynamic markers. We sort of really started to think about actually frailty, the concept of vulnerability to insults, could be mapped in epidemiological studies, looking at the responses to interventions. So very simple ones like a lying and standing blood pressure, or its simple sort of its tilt equivalents. Also things like a dual task test within gait speed, trying to introduce that extra element of a stressor when we are collecting data from our aging twins, so that we can differentiate vulnerability to stressors going forward. So then about sort of, I think it's about 4 years ago, can't remember, COVID is such a big feature, isn't it? We thought about how we could do that in a really fundamental new way. And we pioneered a sort of dynamic testing study, which we call the PREDICT study, which is a collaboration with the health science company Zoe, where we brought people into their usual visits, fasted, but instead of just leaving it there, we then gave them a dietary intervention, which was standardized within the clinic. We measured their response to that dietary intervention over a 6-hour period, using blood spots and also venipuncture all the way through that day. And then they went home and they continued to do specific menu system of meals. And so we could see how they responded to diet changes over time. And I think, you know, this is a short-term intervention. It's not something that's going to majorly change their long-term trajectory, but it's something which really going to understand physiological responses and metabolic responses to these dietary challenges. And so that was jolly sort of successful piece of work. And unsurprisingly, many of you will sort of like anticipate that there is a very variable response to this intervention within individuals. So measures of inflammation, for example, changing over that period of time. And the differences between individuals could be explained to a certain extent by information that we had already within the cohort. So, I mean, ultimately, we've got twins here. We can look at differences in responses between twin pairs where they are genetically identical as much as can be, and even identical twins responded quite differently to foods that they ate, even in the standard diets that we gave within a clinic setting. And so here, this is just a kind of a schemata of this kind of idea that actually, so, you know, we always think of, you know, calories is what derives your metabolic responses, maybe to a meal, and then therefore your weight and so on. But indeed, the actual meal content, which is in a sort of grassy green color here, is really very little in terms of the source of variance of what then defines the response, in this case, to triglycerides at the 6-hour rise. Individual characteristics like your body mass, your clinical biochemistry, sex and age, you know, take account of about a quarter. But more than half is really unexplained, even when we've got multiple different things here measured. And that's why we need to go deeper to look at, see how individual sort of like things such as experiences to that date, physical activity, sleep, all these other measures that we could actually add to this study to really understand that big grey area if we moved into more remote and digital phenotyping of these individuals. Interestingly, you'll see here that genetics and the microbiome actually accounts for quite a lot of individual variability, probably the microbiome more than an individual's genetics itself. So then we come, so now I'm going to move on to my sort of next bit, which is around, you know, COVID came along, didn't it? And I'm sure that many of you will have adapted to the COVID pandemic in a number of ways. In particular, sort of things that we could do in cohort studies massively shifted, didn't they? But also we were thinking, how can we use our expertise and our experience in epidemiology to try and really understand a bit more about this disease in this data poor landscape? And I was particularly interested, as a TwinsUK, it covers the whole of the UK. We've been working on geography in its relation to health. And immediately we could see how we could use that experience to understand how the disease tracks across the UK population if we had a big enough data set. So we had a population in the UK that was extremely thirsty for answers. They're all locked down and so couldn't do anything. So people really wanted to make a difference at this time. So we launched the Zoe COVID study, which was designed by myself because I was looking after people who had COVID and working together with nurses who had COVID, so a big spectrum of disease. We could see that the disease was presenting very differently in different people. We could see what symptoms they were. We popped them into an app, and together with Zoe then actually developed that technology to then really give it out to the whole of the UK population. We've got 4.7 million people signing up, mostly in the UK, but also in Sweden and the USA. And partly that was because of really good media relations that we developed over the course of our research program, which meant that we could get the message out there to get people to join. And so, you know, I'm sure that many of you across the world don't necessarily know what impact we made, but really we had a pretty good impact within the UK population, understanding the disease, and also worldwide. We were responsible for both UK and WHO adding in anosmia to the guidance for testing. We were responsible for understanding the importance of delirium in frail older adults in particular. And of course, that benefited from this huge interest from the population. Daily logging of the symptoms enabled us to then actually map how this was working across the UK. So before we had really good testing data, we could see how the disease was spreading and tell the government about hotspots of areas where they needed to potentially introduce further lockdown measures, in particular Leicester from quite early on in the pandemic. But of course, that required a really active pool of participants. It required a huge engagement campaign. So we really were ramping up how we engage with our participants within the cohort, really on a real-time basis, feeding them back research insights very regularly to keep them engaged, and constantly adapting the app so that it keeps it being interesting. And we've actually sustained, we've still got about 600,000 people reporting every week after 2.5 years now. And now they're not just reporting on COVID, they're reporting on a whole range of different symptoms to us. So here's some of the impacts. I sort of mentioned some of them: the sense of smell, delirium, but also long COVID. We've actually had about 40 peer-reviewed journals. I mean, I'm almost losing track, but we have had some really big impact papers as well from that. And because of this daily hotspot sort of and other sort of insights that we could get, particularly using case control analysis from this, within this really very large data set to answer specific clinical and government impactful questions, we had a direct line to sort of some of the key decision makers in the UK government and abroad. And in the meantime, TwinsUK was able to take up a vanguard role within something new that was set up within cohort studies in the UK called the National Core Studies, which was set up by our Chief Medical Officer, Patrick Vallance. And I'll tell you a bit more about that in a moment. So here's another, which is one of the other things just to pick out. So this is a case control study where we looked at who gets post-vaccination infection, and if they get post-vaccination infection, what does it look like? And we could see, I mean, this particular slide is showing that individuals who had post-vaccination infection were much less likely to have long COVID than otherwise. So really important, actually, the first piece of evidence that says that we have got a treatment to prevent long COVID. And that's since been replicated by quite a few others now. Yeah, so apps. So apps, and how do we use this digital technology within population studies going forward? Well, apps can tell you the picture on the ground. So here's a nice picture from early in the pandemic showing that you know, something simple as healthcare workers, do they have access to PPE, personal protective equipment? And we can see that, you know, within hospitals, pretty good, nice and blue there. But within the elderly nursing home sector, real difficulties, quite a lot of people in that yellow band saying they don't have access to PPE. We could immediately feed that back through and help policymakers change things because of the fact that we could map things on a population scale geographically. But also from individual point of view, this graph on the right here is a single individual. They're reporting on the app about the symptoms they got when they got COVID. So you can see that there's a sort of diamond there at the top, which indicates this person tested positive for COVID, I think on the 30th of March 2020. And then, you know, they had fatigue right all the way across, but the fever only lasts a short period of time, sort of maybe just over a week. And the headache lasts a little bit longer, but then other symptoms coming out, such as skin symptoms, muscle pain symptoms, and this fatigue and cough that really lasted for a long time. This is one such example, but you know, we've used this massive data set to really understand how long COVID can come in different symptom clusters and describe them carefully for the world's population. So back to the cohort study, TwinsUK, that this all started from. Well, we didn't stand still either, and like many other cohorts across the world, we're sort of really interested in using this time to map the pandemic. So we produced something very quickly right at the beginning to try and understand the personal experience of COVID, and then we shared that with the longitudinal population studies community within the UK through the Wellcome Trust, who's our main funder. Putting together Generation Scotland, ALSPAC, other CLS studies, sort of like the birth cohorts within the UK. So we share this so we can do it across multiple different studies. And very quickly, that sort of then generated what I'm going to show you in the next slide, which is the National Core Studies. But also, we weren't going to stand still with our twins within London. We had to stop twin visits, but that didn't mean to say that we needed to stop biological sampling. So we went out into the field and have collected samples from individuals, which we've actually now used, for example, to look at epigenetic aging across the pandemic. And we can see that everybody's aging more across the pandemic than we expect but we don't see differences between COVID and non-COVID, thankfully. But we've looked at other things such as T-cell response, immune response, and so on, with biosampling happening within the cohort. So it's essential to have these cohorts to be really prepared to meet the challenges of pandemics as they come through. So this was just a couple of papers. So the serology then, with the national core studies as well, we then used our experience and expertise in doing that kind of data collection within the pandemic scenario to then roll out mass data collections of, I think it was 50,000 people that are in cohort, longitudinal cohort studies, taking serology samples so we can map them across the pandemic in relation to their disease and also vaccination. So this is just some of the people that that we've been involved with in terms of the national core studies, and we're able therefore to sort of look at the relationship between COVID pandemic and many other aspects. So just coming to the end now, so the question about the future, what's the legacy around all this work? Well, how could we do experimentation in cohorts, either from a point of view of looking at natural experiments and how they play out with time series of data collection across individual exposures, or relationships between what then we actually do as interventions to look and see how things change in terms of individual responses using multi-omics. We've got a new ability to track lived experience over time thanks to digital technologies. We've got massive new engaged populations to sample from and to scale to. And we've got highly engaged cohorts like TwinsUK to continue to test variability to challenges going forward. So that's what I put out to you, whether or not that could be done on a worldwide scale. So thank you.